I think we can get going now. So welcome everyone. I'm Leonie Coyetin, the Industry Development Manager with the Macadamia Society. And it is an unusual event for us today. We're having a virtual field walk. We're going to be seeing presentations and in the field research trials from two projects, the breeding project that we have, and also what we call tree intensification, which is a follow-on from the small tree high productivity project. So a number of researchers are online and they're going to be giving you an overview of what they've found, but also what that research means for you as a grower in the field. So I won't delay anymore and I will pass over on to Mo. So Mo, are you there?
Yes, I'm here.
So are you able to share your screen with everyone?
Sure. Hello, everyone. I'm Mobashwer. I'm a Macadamia breeder here at NAMBO. Today, I'm going to present about an update about our rootstock trial, which is a part of AS 18,000. The main objective of this project is to identify rootstocks that can reduce the plant size, also increase precocity, and also increase yield efficiency. And we are also investigating if we can find rootstock have an effect on reducing size and increase precocity, what mechanism is involved with. We are working with Christian Beveridge and Lindsay Shaw to find out if there is any mechanism we can explore. So this trial we have planted in 2017, in April 2017, and we have included 24 different type of rootstocks. And out of those 24, we try to use 5 different type of rootstocks. We have included wild species, included some dwarf we identified from our breeding trial. We included standard rootstocks, H2 and Biomon, some cultivars which we identified from our previous rootstock study, and also these are some wild species I told you before. So now I'm moving forward to show you some of the results. We identified that this is just 4 years of age. Last year we collected all data, and we identified that yes, rootstock have some significant effect on reducing plant size and precocity. This is really preliminary data, so possibly the result may be different after 4 or 5 years. Just to show you some update, I'm going to show you some of the precocious rootstock we identified here. One of the precocious rootstocks we identified here. So before showing that rootstocks, I'm going to show you one rootstock we used as a H2 seedling. So you can see this is one of the H2 seedling we have used as a standard of our rootstock trial, and this is a grafted tree. So in this trial, we have planted grafted and ungrafted tree side by side to identify if there is any correlation, any association of rootstock, ungrafted rootstock with the grafted scion, if rootstock is transmitting trace to the scion. You can look at the trees. This 741 grafted on H2 is vigorous, and normal 741 looks like it's a bit upward type. Now we are moving to another rootstock we have used in our study. This one. So this one really showed its performance to increase the precocity of 741. This started flowering at age 3, and last year, up to last year, it produced 4 kilograms per tree. So this is the rootstock we called BHI1, and this is the grafted 741 onto BHI1. You can also see the variation in structure. You can see this is a bit open, just look like this one. But when we looked at the H2, that is a bit upward type. So it's until today, although this is preliminary trial, but until today we can see that rootstock have an effect on tree size and tree structure and precocity. Now I'm moving to another tree. to compare how if rootstock reduce plant size. We have identified a few rootstocks which can contribute to reduce the scion growth, and one of those is a Jansenii seedling, and this is one of the Jansenii. It is 5 years old now. You see it is small tree, and this is the grafted 741 onto Jansenii. It reduced the plant size dramatically. If you compare with another tree there behind me, those are really 3 times vigorous than this one, maybe 4 times. So this is our findings till today, but we didn't know how it is happening. One thing we have noticed here, if you look at here, there could be some graft incompatibility. That could be the reason of this reducing size, but this thing didn't happen in other trees. So actually we don't know until today, but Lindsay Shaw is helping us what molecular mechanism is happening in this rootstock scion interaction. Thank you very much. If you have any question, please don't hesitate to ask me.
Great, thank you, Mo, for that. Are there any questions from any of the participants? Please feel free to type them into the chat box there. We've got a couple of minutes. And any of those questions that we don't get to during the webinar, we will email out answers to, as Leonie mentioned previous. So Grant Thorpe has just sent a question through. Mo, how many trees showed graft incompatibility?
Just actually this one only we have seen, but this one we can see the maximum of incompatibility, but we didn't see that much in other trees.
Okay.
And yep.
And there's another question here. What was the name of each rootstock?
So the previous one I showed you, that is one of the selection from our breeding trials, from first generation breeding trials. So we gave it a name BHI1. This is not commercially available, and that one we have selected. The main reason is that one have high breeding value for selection index. So selection index composed of all the traits, all the beneficial traits we are looking for. And based on that, we have selected that one as a parents for our breeding. And we also wanted to test them as a rootstock.
Great.
And this one, another one is Jansenii. It's a wild extensions. So one of the wild extensions of Macadamia Jansenii that reduced the size dramatically.
Mo, did you want to just touch on the recent coffee webinar that you have done, and that I can send a link out to the participants to watch that recording?
Yeah, sure. Yeah, I can.
Yeah. So Mo just recently did a coffee research webinar update, which I'll include in the email out to everybody. It's a 45-minute presentation that goes into detail around all the great research that Mo and his team are doing. We have another question here. Are you measuring water use and measuring gas exchange?
That's we didn't have planned to do it in this project, but we have a couple of students working with us. One is an MPhil student. Hopefully, she'll be converted to PhD. Yes, she wanted to do those things. She wanted to look at the hydraulic conductivity and gas exchange, and she also wanted to look at the stomata size and density. We have done a little experiment on that using 3 different rootstocks, and we have identified that rootstock have a significant effect on stomata density. So that's why if she got time, she will be doing that thing as well.
Great. All right, well, we might, there's 2 more questions and then we'll move on to the next agenda item. Leonie says, so Mo, these aren't publicly available to growers, But from this
No.
trial, we have a chance to inform further breeding work?
No, these are not publicly available.
Yeah, but being used to inform further breeding work?
Yeah.
And Andrew Pearce asked how many different scion varieties were used?
The main reason we have used only single scion, that is HAES741. We want, because that is non-precocious, we know that. So that's the main purpose, one of the main purpose. If it can reduce, it can increase the precocity. So only one scion variety we have used in this trial.
All right, well, thank you, Mo. We'll move on to the next agenda item, but if you've got any further questions for Mo and the breeding team, please feel free to type them in and we'll answer those in the follow-up communications. All right, so the next agenda item is a pre-recorded video that the Bundaberg AS18000 DAF team have done, which is the phase 2 macadamia planting systems trial, which is based at Mac Farm Management. So this trial is part of the new AS18000 program of tree intensification, which is a flow-on, which Leonie mentioned, from the Small Tree Productivity Project. So I'm just going to share that video now.
Welcome to Mac Farm Management. We are here to show you our second planting systems trial. We have 2 planting systems trials. These are research trials in which we compare the effect of different components like scion variety and tree structure on productivity. Our first planting systems trial is on the Bundaberg research station, and there we compared the effect of planting density, tree training, and scion variety on productivity. From this first planting systems trial, we have learned that high early macadamia nut and shell yields can be achieved using intensive planting systems. We are very well aware that we do not know how these high-performing systems are going to perform later in the orchard life. So we set out to design a trial likely to be highly productive and able to be managed commercially in the long term. So here we are in our second generation planting systems trial. The trial was planted in April 2020 and is now 2 years old. We have 2 driving points in this new trial. The first one is high yield early in the life of the orchard, and secondly, high overall yields. The first aspect that we looked at is tree density. We have settled on a tree density of 5 meters by 3 meters, which gives you 667 trees per hectare. This lies between our high density and medium density plantings of our first trial, where the high density was 5 meters by 2 meters and the medium densities is 6 meters by 3 meters. Secondly, after very careful consideration, we chose 3 scion varieties. The first obvious focus was precocity to get our early yields, and the second criteria is tree size. Here we stand at an A16 tree. This tree is fairly precocious and it is known to have a high yield efficiency. That means a high nut and shell compared to canopy size. It is also planted quite widely in the Bundaberg area. A203, although not a small tree, is extremely precocious. In our first planting systems trial, we got up to more than 100 raised seams when the trees were 2.5 years old, and at 3 years old they had an average of 2.4 kilograms of nuts in shell per tree. So we decided that this tree had to be included in this new trial as well. P is one of the newly released varieties from the industry breeding program. It is reported to be small and relatively precocious. The next aspect that we looked at was tree structure. we decided on 2 tree structures in this trial. The first one is the vase structure, because previous tree structure research in macadamia has identified that vase canopies had a higher yield efficiency than other treatments. We are aiming to select 4 evenly spaced leaders on each tree that are spreading upwards and outwards, thus creating the vase structure into the alleyway. From a practical point of view, this is challenging in Bundaberg due to the strong and consistent prevailing winds. The second tree structure that we have got in this trial we call our complex tree. Due to the importance of early flowering for early yield, it is important to have a certain amount of canopy complexity or branching to bear these early flowers and fruit. We created this canopy complexity by tipping shoot axes that grow more than 30 or 40 centimeters without branching, like those 2 that have just been tipped. We are now happy with the complexity that we have achieved, and will end this tipping now, just at the start of our first cropping season. As you can see, the tree is quite complex at this stage. To manage the vigour in this trial, we have looked at the following. Firstly, we use varieties that produce small trees. Secondly, the soil at this site has got a low to moderate fertility. And thirdly, we are going to introduce a plant growth regulated treatment to limit the vegetative growth. This will be applied once the trees have filled their allocated space and we need to slow their growth. So this was a quick overview of this trial. It is still young, and although a few individual trees have carried fruit this year, our first cropping season will be 2022-23, in which we will commence to measure the productivity of these trees. Finally, we've got our demonstration block. This is planted adjacent to our trial at the same planting density of 5 meters by 3 meters. So also 667 trees per hectare. Double rows of the same 3 varieties, A203, A16 and P, are planted in this block. The project team is working with Macfarm management to collect orchard establishment and management costs and production data. This data will be used to better understand the economics of intensive macadamia plantings and how they compare with conventional plantings.
Great. So are there any questions regarding the phase 2 planting systems trial video that we just shared? So Leonie asks, Carola, we have Carola on the line, who was a lovely lady in the video, who's part of our Bundaberg research team. Leonie asks, what is the plant growth regulator you are intending to trial?
Leonie, at the moment we're trialing 2. We're trialing uniconazole as well as paclobutrazole. And it depends a little bit on the results that we get in our pre-trials as to which one we will use in the end.
I should mention that as part of the upcoming MAC group meetings, we hope to have a field day at this MAC farm site. So those that are in the local Bundaberg area or planning to travel up to a MAC group meeting in that area, you're more than welcome to come along, and there'll be notices sent out through AMS in due course around that. So you can see the trial in the flesh, which will be good. We have another question, Carola. What is the name of the last lot of tree varieties that was mentioned?
Well, so in all of those trees that I showed you, we have got A16, A203 and P.
And Andrew Pearce has a question. Why stop with the tipping on the complexity trial? Would it be better to keep that going and keep tree size small and complex?
Andrew, we're quite happy with the complexity at this stage, but it is an ongoing process, and it wasn't pre-decided exactly when we will stop with a frequent pruning, but yeah, we'll see how we go, how the trees are going. At the moment, they look pretty good for the next flowering, and we might leave it at this stage but still do more frequent pruning in the future.
We have another question. What other practices will they be using to reduce canopy volume, i.e. mechanical hedging?
No, we're not planning to use mechanical hedging at this point. In the future, we might do more selective pruning. And well, we are hoping that by using the plant growth regulators that we can reduce or keep the canopy volume pretty small in that way.
Would you like to mention, Carola, just the soil type chosen?
Yeah, of course. Yeah, of course. The soil type. The trial is planted on a fairly bad soil. It's not really very fertile, and also, yeah, by pruning, we will have to prune the trees once they have filled their space. But this should take longer than, for instance, at our other planting systems trial, which is planted on really very fertile red soil.
I'll be sending out a article that John Wilkie and the team put together, which covers the findings of the previous small tree project in the planting systems trial phase 1, and how those learnings have been brought forward into this new planting. So I'll be sending that out for those that are interested in reading a bit more. Andrew asked, did you say what rootstock, sorry?
The rootstock is H2 on all of them.
All right. Well, we'll pass over to the breeding team now if there's no more questions. We're going to start with a short video of Bruce in one of the blocks, which I'm going to play shortly, and then we'll go across live to Bruce in another block. So I'll just set that up now. But thank you, Carola, and yeah, I'll share this video now.
Two, one.
Yep. Good afternoon. My name's Bruce Topp. I'm the leader of the Macadamia breeding program, and I work for the University of Queensland for QDAF. I'm here today at the Maroochy Research Facility, and it's a great opportunity to talk to you a little bit about what's happening in the breeding program. I'm located here today in what we call our high breeding value block, and this is where we do the pollinations each year in order to produce the new segregating populations of macadamia seedlings. And this tree, for example, was one that we selected from the first generation of breeding, and it has been used as a parent to create the second generation. Further down this block, we have second generation parent trees that we are currently using to create the third generation. And so it's a continual process where each generation we're looking at making improvement in new cultivars that are going to be released to industry. Okay, great. Oh yes, well, so here we are. We're in still at Maroochy Research Station, and I'm over in a different block with Grant and Mo at the moment, and we just weren't quite sure of the technologies, and hence the video for that previous block. We we weren't sure if we were going to lose you when we moved one block to the other. Anyway, that's been done, and we're over in this second block of trees, which are quite different from that high breeding value block that I just was in and was talking to you from. But before I tell you about this new block that we're in, I did want to mention that we're creating a lot of new seedling populations through the hybridizations that we do over in that block that you just saw the video from. Now, when we get these hybrid seed, we germinate them here at Nambour and also up at the research station at Bundaberg. And then in the past, we've been planting them out at research stations. So we've got some planted here at Nambour, some planted at the QDAF research station at Bundaberg, and we're planting others this year down at the Wollongbar research station of the DPI, New South Wales DPI. But very importantly, we also plant these seedling populations on grower properties. And I want to make a point of this because the grower cooperators that allow us to plant these progeny field trials on their properties are an integral part of the breeding program, and we couldn't get what we get done without their assistance. Now, in that regard, these hybrid seed that we've just created last spring, and we're just in the process of harvesting now, we're looking for a place to plant those seedlings out close to the research station so as to reduce the travel time when we have students working on those projects. So if anyone's listening, this is a plug to see if there's anyone in the Nambour district, maybe down around Glasshouse, then we'd be very interested to hear from you if you have some land available for us to plant a progeny trial at your place in about 12 months' time. Okay, so that's the plug over and done with, and I'll talk to you about this this new block. This is quite different from the block we were in previously. Previously, we were looking at the progeny trees and the elite selections that were planted at about 8 metres by 4 metres, the industry standard. Here, and I think Grant's just panning around to show you, they're much, much higher density. The trees you're looking at the moment were planted in 2020, in November 2020. The seed were actually on the tree in March 2020. So these are very young trees, and they're planted 0.5 metre apart. Just to show you what they look like when they get a bit older, we'll move down this way. And this next lot of trees that we're looking at were planted in 2018, so November 2018. And these trees are at the same density. They're planted at 0.5 metre apart. And the idea behind them is to speed up the breeding program. I talked to you before about, I kept talking about generations, how we, first generation, and how now we're creating the third generation. So there's two things that are really important. There's a lot of things that are important in breeding, but two of the very important ones are the length of the generation cycle, and so how many generations you can get through, and the size of the populations that you're able to evaluate. Now, what we're looking to do is we're looking to make stepwise progress each generation of breeding that we go through. And therefore, the quicker we can go through one generation to the next, the greater the genetic gain on a per annum basis. And one of the ways that we're doing that is we're selecting for more and more precocious seedlings. And so, for example, in, say, 10 years ago, 20 years ago, if you harvested a nut from one of our progeny and you grew it up as a seedling, it would take, on average, about 7 years for that seedling tree to produce nuts. In this particular block of trees that were planted in 2018, we've been able to go from nut to nut, from harvesting a nut on a tree, germinating it, and then on the resulting seedling, see nuts on that. We've been able to do that in 3 years. So that's an incredible improvement in the precocity, and it just means we can speed up the whole breeding progress by reducing the generation length. The other thing I mentioned that's really important is the population size. And the more seedlings you have a look at, the more seedlings that you evaluate, then the greater the chance you have of finding those elite ones that the industry needs. And at this high density, obviously we're able to look at and evaluate a much larger number of seedlings for a given cost. This particular high density planting, obviously you couldn't grow these trees to 20 years old. It would just be a crazy jungle. But what we're intending to do with these trees is to push them all out at 5 years of age. And so what we're doing is we will go through these trees this year when they're 3 years old. We've had 5% of the trees have produced fruit. And next year, when they're 4 years old, we expect that to be about 20%. And once we get to about 30 to 40% of the trees that are fruiting in year 5, we will then cut out all the rest. So the ones that are left, we know are highly precocious. We'll measure the kernel recovery on them, and we'll only keep those with a high kernel recovery. And then once we've narrowed that down further, so we've got high precocity, we've got high kernel recovery, then we'll use the work of one of our students, Dr. Katie O'Connor, who studied genomic selection in our macadamia breeding program. And what we will do is we'll go through those remaining seedlings, the ones that are left after they've been selected for precocity and selected for high kernel recovery, and we will take DNA samples from the leaves and use the genomic selection model that Katie has developed and that other members in our team are refining to select out those that are predicted to have high nut and shell yield. And those elite selections we will then put out on grower property for test. So it's a way of speeding up the whole process by using some of the modern DNA technologies. We would like to do the DNA testing at the seedling stage before we even planted anything out in the field. But unfortunately, the cost of the genomic testing is not cheap enough for us to be able to do that. But we will keep an eye on that and see how that progresses in future. With any of this testing, the prediction of the performance is just that. It's a prediction, and it always has to be validated by trials on grower properties in many different regions. And that's where our close linkage with DAF's regional variety trials is so important.
Great, thank you, Bruce. Do we have any questions for Bruce? Hey, Bruce, do you mind just holding the iPad still? There's a bit of shuffling of the mic.
Oh, my apologies, Bridie. Yep.
All good. Do you have any replication, or is every seedling unique?
Yeah, look, that's a great question. We have replication of family. Every seedling is unique. We have replication of families, and so what we do is we like if we've got if in this block that I'm standing in we had a particular family and we had 100 seedlings in that family, we wouldn't just plant them all in one plot. We divide them into several different plots so that we've got randomization and replication of the family, although we don't have replication of the individual seedlings. To take that one step further, one of our associates in our breeding program, Dr. Craig Hardner, is working on this idea of how we can improve our breeding strategy. And one of the things that he's investigating is if we do a trade-off between the number of seedlings that we evaluate and replication of those seedlings, how will it affect our genetic gain? Obviously, the more, as I explained before, the more seedlings that you look at, the better chance you have of finding that elite one or several that you really want. However, if you have replication of the seedlings and therefore are not able to plant out as many genotypes, you will have a more accurate measure of the traits that you're evaluating. And so what Craig's doing is assessing whether that trade-off for the number of seedlings that you evaluate versus the accuracy improvement is worthwhile in terms of the overall genetic gain.
Great. Are there any further questions for Bruce? How has the KR cut-off been established yet? Has the KR cut-off been established yet? Sorry.
Yeah, no. In our regular breeding program, this is a novel approach. This high density planting and using genomic selection. This is a novel approach for us. In our regular breeding program, the kernel recovery is not set at a definite threshold. What happens is we use something called a selection index, and Mo referred to that when he's talking about some of the best performing rootstocks that are in our trial here. The selection index, what it does, it combines all the different traits that we're looking at. So nut and shell yield, kernel recovery, tree size, they're the three big ones, precocity, but there's a whole range of other traits. And what it does, the selection index, is it gives a weighting to each trait so that we get a value for each new genotype that's based not just on a cutoff threshold for certain traits, but we will give and take. So, for example, if something had a kernel recovery of 38%, and something else had a kernel recovery of 42%, we may end up selecting both of them if the nut and shell yield of the 38% one was higher than the one with 42%. And this selection index is a mathematical equation that allows us to make those trade-offs in selection.
Great. Do we have any further questions before we hand over to Inigo? Is precocity associated with mature productivity?
Yeah, and this has been, there's a debate always amongst, within the industry about this, and I don't think it's been settled definitely yet. You know, there's a grower that I know refers to the racehorses and the stayers, and the racehorses that he's talking about are the ones that are precocious and produce high yields early, but don't have the staying power, whereas the stayers are ones that have a slow and steady increase in yield, but in the long term they produce well and continue to produce well. Obviously what we would like is something that has both, that is precocious to start off with, but that in the long term does have a good mature yield. Now, as to the question about whether you necessarily trade one for the other, I think that, you know, what we need to think about is that it's inputs and outputs. And so if you do have something that's precocious, then there's a lot more being taken out of that tree early on in its life, and you can't treat it the same as a tree that's producing at a much lower level early in its life. And I think that's been shown with some growers. Some growers will find that things like precocious things like A4 or A16, they will say, oh, they're good early on, but then they die off later on. But there's other growers I know of that swear by them and say that with the right management, the precocious ones can continue to produce into mature age.
All right, thanks, Bruce. We'll answer those further questions that people have typed in the email post the webinar. Sorry, we're going to have to move on to Inigo. We're running out of time. But thank you, Bruce, and yeah, that was really great, really interesting.
Thanks, Brady.
So now I'd like to introduce Inigo Uzmendi. He's a modelling researcher based with QDAF, and he's going to be presenting some of the macadamia modelling work as part of the AS 18,000 program.
Thank you, Brady. Yeah, as Brady mentioned, my name is Inigo Uzmendi, and I'm based in Brisbane, working at QDAF. This work is part of the National Tree Crop Intensification in Horticulture program, and today I would like to show you how we can use modeling to show how we can use modeling to understand better how the macadamias and the fruit of macadamias grow. Probably you have heard about modeling before. Usually modeling is used for predicting likely outcomes of different scenarios. Sometimes they are used as decision support tools. Some of them have been used for scheduling irrigation or for nutrient budgeting. And one of the advantages of the models is that once we have it, we can run simulations, or we can run it and get results very quickly, like in minutes or hours, in comparison with a field experiment that will take a few years usually. On the other hand, models usually focus on a very simplified system. They target some aspects of the plant, so they don't simulate the full complexity of the plant. But because we can focus on some aspects, it can help us.
Sorry, Inigo, are you going to share your screen?
You don't see my screen?
Your presentation?
Yeah, I thought I was sharing it. Wait a moment. You don't see it. Okay. So what's wrong? Yeah, I forgot sharing the screen, sorry. Okay, now you see it?
Yes, thank you.
Okay, well, it doesn't matter because what I have been talking is pretty much about the title, and that's what you get. So I was saying that, yeah, with the modeling, we can concentrate better in several aspects and understand better how the plant physiology works. When I'm talking about modeling, talking about virtual plants, which are simulations models that allows us to replicate the three-dimensional structure of the plant and how it grows. And we can get in our computer, this is my computer in my office, we have you can see in the right side screen, or the photo of the screen, the code, the mathematical functions and software that we use for creating the model. And in the left screen, you can see the simulation or visualization of the plant, in this case, one macadamia tree. The first thing that we need when we want to simulate the growth of the macadamia is to get the architecture of the macadamia. This is called digitizing. You can see in the right side macadamia in the field, and this instrument is part of a digitizer, which uses a magnetic field, and that is used with our specific instruments to collect the position of every organ, in this case, every internode in the tree. Once we get all this information recorded, we can visualize that in a computer, which is what you can see in the right side of the screen now. And this is work that has been done, for example, at DAF by Neil White, Graham Bignell, and Stacey Griffin. So using this organ size and position as input, we can model the tree physiology and growth. In the image, in the picture, we can see with brown color is the trunk and the stems. Then with dark green is the color of the leaves that we had at the beginning of the simulation. And in yellow, you can see the fruit, Macadamia racines. And the new growth that has been simulated is color with a green light. In order to have these simulations, we need as well the potential organ growth that has to be measured in the field. And we use this to simulate the growth with different external factors and to understand better plant physiology, with the final aim of improving the orchard productivity and sustainability. I have put here a couple of references, just in case you want to know more about this. The first one is a little bit more technical, and the second one, you might have read it, is was published in summer 2021 in the Association of Macadamia, the Australian Macadamia Society news bulletin. So when we speak about external factors, we are talking about, for example, the phenology, the dates of flowering and bud break, or the dates of different flashes. And we talk as well about the light that is received by each individual leaf during the whole day. The internal plant processes that we are simulating are photosynthesis, that is the production of carbohydrates, and then how these carbohydrates are distributed within the plant, according to or following some organ competition. This simulation is usually performed for one growing season, starting at flowering, or probably better is fruit set, up to harvest. So here I have a video to show you simulation that we did with Macadamia. There's a whole canopy behind, but here he is showing you a zoom or a small part of the canopy so you can see the individual organs. So what we are going to see is the growth during one growing season, and you will see that the fruits that are in yellow will slowly grow, and at some point the dormant buds that now you can see as brown will become light green, and they will grow one flash, and then they will go again dormant. So this is the simulation. The fruit is starting to grow. At the beginning it's not that clear, now it's getting more clear. Now we have the first flash. It goes, then the bud becomes dormant. Wait for a few days, and then it will start the second flash. And during all this time, the growth of the leaves and the stems is competing with the growth of the fruit. And this is simulated by the model. So what we did is, we used this model to simulate how the growth of the different parts of the tree will happen or will occur, considering that the distribution of carbohydrates is happening at different levels. That is, that the whole tree is sharing all the carbohydrates with all the organs. Then we compare that with the whole limbs growing from the trunk, sharing the carbon, and then carbon share within the 2-year-old branch, and carbon share within the 1-year branch or branchlet. And what we observe is that looking to this flat autonomy, considering that the carbohydrates were shared within individual 2-year-old branches, showed the most realistic behavior, or the one that matched closer the data that we collected in the field. One consequence of this is that we saw that we have longer shoots and bigger leaves and fruits in the top and external parts of the canopy. That's due to having more light. So with more light, we have more photosynthesis and create more carbohydrates in those parts. But these carbohydrates are shared within the branch, within the 2-year-old branch. It's not just the light that is receiving the organ, it's the light that is received within the whole branch. And definitely we saw that not all carbohydrates are available for the whole plant, because if we will have that, then all the organs will grow the same in all the parts of the plant. Which are the implications of this branch autonomy? How that could be useful for growers and scientists? Well, first of all, this indicates that we can consider smaller parts rather than the whole canopy when we want to study carbohydrates. Because there are differences within the canopy, it's clear that it will be interesting or will be useful to have a good light distribution to avoid zones that are not getting enough light and not getting enough production. However, we have to be careful because carbohydrate distribution and branch autonomy is not very well known in plants, because it's difficult to measure it, and then we haven't had too many experiments in this area. So we use these simulations, as I said before, for understanding the macadamia tree physiology and for defining as well innovative orchard design and management practices. For example, once we have the model, one tree, we can put several trees together and simulate an orchard environment. For example, this is a picture of another experiment in which we put 9 trees together. and look to the tree in the middle. But this is another subject for another day. Today was just a brief introduction of what we are doing in modeling and how we are working with it. Thank you very much. Any questions? Thank you for the attention.
Great, thank you, Inigo. We have one question from Leonie. Have you started to model different cultivars? Can you take this and rootstock into consideration in the model?
Okay, at this point, at the beginning, we are simulating a generic Macadamia tree with some data, not the architecture, I think that it was from 741, but we are not defining that for individual cultivars at this point. And simulating the effects that rootstock could have on the Macadamia, it has been done in other models, for example, in a peach model in California, but we haven't tried that in our model. In fact, in our model, we are not considering the root system at this stage. So, but that's something that could be used at some point.
Great, thank you. Any further questions for Inigo before I hand over to Leonie to wrap things up? All right, thank you, Inigo. That was great.
Thank you.
Leonie, over to you.
Sorry, I was on mute, as we all so often are. Thanks to all the researchers that shared today. It was an important look for growers and for other industry members to understand that there's so much work being done by the Macadamia levy on your behalf, looking at how we can take the industry forward. So we might not have that many on-farm outcomes from what you've seen, but what you've seen today is the groundwork for our industry expanding and growing over the next few years. So if you've got any questions that weren't answered today, the team are always happy to be engaged and involved with you. And like Bridie said, we will have more opportunity at our MAC group meetings for you to be able to interact with some of the researchers. So thank you so much for logging in today and taking a look at some of the work that your levy is doing. We hope you have a good day and a good season.